graduate class that I've taught in the past. So this will be extremely expedited, and we'll discuss just general aspects of some of these molecular and cellular mechanisms, just to give some insight into some of the regulatory components involved. So just understand as I go in, this process, as Dr. Roberts could attest, is extremely complicated, and there are still things that we don't know about molecular and cellular mechanisms of hypertrophy, and many that have yet to be identified. So what I'll be talking about today are just some of the basic components that we have. We've got enough data on to be able to construct a bit of a picture, if you will, to have somewhat of a general idea of what's involved in this process. So,
hey, Darryn, can I interrupt you for 1 second?
Yeah.
I just want to tell the audience that if they have any questions, use the Q&A box at the bottom. Don't use the chat box. So again, use the Q&A box if you have any questions. So, okay, go on, Darryn.
All right. So in terms of the overview of the presentation, so just to kind of help compartmentalize things, I've got it set up here into groupings where, we'll start off talking about just kind of an instigation scenario where these are some of the components that actually help initiate, if you will, instigate the process for muscle protein synthesis that ends up, at some point in time arriving at muscle hypertrophy. And so some of these that we'll touch on are mechanical stimuli, mechanotransduction, hormonal, nutritional, and then we'll talk about an interactive process, which is just basically just how the instigation process is actually linked to the gene expression transcriptive process. And that's just, for the most part, linked to enzymatic interactions, protein to protein interactions. So again, that's the interactive or the linkage component, if you will, from the stimulus to what ends up arriving within the nucleus to upregulate gene expression. And then we'll talk just basically about transcription itself, gene expression, some regulation of gene expression based on messenger RNA regulation, and then the process of translation, where first of all we synthesize polypeptides, and then those guys end up being processed into fully active biologically represented proteins. And then I want to touch a little bit there on a cooperative component there, which is involved in the myogenic activation of satellite cells, because we know that data showing that muscle hypertrophy can certainly be augmented, if you will, based on the contribution of satellite cell activation. And then I'll finish up just with a short statement about the degradation process relative to how muscle degradation can actually impede hypertrophy. And obviously this is not something that we would want to find ourselves within, at least for any extended length of time. And there are many aspects other than just myostatin and reactive oxygen species that seem to play a role. And so, but anyway, so again, this is just something we'll touch on going forward. So again, the first process, as I mentioned, is just the instigative scenario. And just to give you a just very brief, and if I can real quick, you're going to see as we go through here, almost all these slides are all pictures of cell signaling pathways and such. There's not going to be really any text on these slides, because I think that just putting words on the slide really doesn't do that much, other than just, it's just information for you to read, and at least the pictures. And I'm just going to expedite the pictures because most of them are very busy, but at least it can give you just a very graphic pictorial display of how some of these processes work. And it's not necessarily, understanding every little component on the screen is really not overly important for us. But I think just having a good understanding of the general process per se is what we want. So mechanotransduction this is the process simply just by which stimulation of muscle contraction, if you will, the tug and the pull and the strain on the sarcolemma or the cell membrane of muscle is going to, through the extracellular matrix, is going to end up stimulating some these proteins and some others. The key proteins seem to be what's called the integrins. And so, muscle contraction via exercise, particularly heavy load resistance training, and it seems to be, the higher the load also is going to accompany a higher amount of eccentric component as well. So that stimulates these mechanoreceptive proteins. That then activates a cell transduction pathway within the sarcoplasm that ends up arriving within DNA and helping to upregulate various muscle-specific genes that end up being important for muscle hypertrophy going forward. All right, so just understand that mechanotransduction is an extremely important component for muscle hypertrophy, because think about it this way. Nutrition is important, yes, but we can eat all we want and not resistance exercise, and we're not going to have muscle hypertrophy. So adipocyte hypertrophy, but not muscle hypertrophy. And the other thing is relative to hormones. Let's think out, from the standpoint that hormones are important, but if we talk about supraphysiological doses of anabolic related hormones that someone might take, if they're not doing resistance exercise and calling into play this mechanotransductive component, then there's not going to be much muscle hypertrophy going there as well. So this right now, this scenario right now, guys, cannot be overlooked in terms of the relative importance of muscle hypertrophy. So this obviously is very, very busy, but all I want to do is just to show just some of the hormonal or growth factor in induced instigation. Of course, here's the components here relative to mechanotransduction, and of course, it ends up being combined with our hormonal components. But here we got human growth hormone, for instance, insulin-like growth factor 1, hepatocyte growth factor, FGF, which are particularly HGF is involved in some extent relative to the role of the myogenic cascade. But in this particular case, it shows the integration of the hormonal cascade within the cell itself on these cell signaling pathways that end up arriving within DNA to upregulate the expression of various muscle-specific genes. So, we've got these hormones here, obviously, these being peptide-based hormones that bind to their extracellular receptors. We could have testosterone over here, for instance, that is because of its lipophilicity, ends up being able to bypass an extracellular receptor and bind to its intracellular receptor. But nevertheless, we've got the impact of this hormone as well that's having the relative same outcome on muscle-specific gene expression as some of these other hormones as well. So we need to take into consideration the hormonal factor as well. And of course, we know that with exercise and we can get some transient increases in some of these hormones that some data say that they really don't play a role in muscle hypertrophy going forward. But that being said, the contribution of these hormones to the integrity of skeletal muscle physiology is certainly something that can't be overlooked also. So if we talk about nutritional induced instigation, of course, this is something that, is within the International Society of Sport Nutrition. Not that we don't hold these other components near and dear to our heart, but we certainly do within the ICSA, because this is something that we know that we can potentially manipulate through our nutritional strategy. And so we know that both glucose and amino acids can generate an insulin response, obviously glucose, but some amino acids, leucine and some of the other essential amino acids, are insulin secretagogues. So they can generate an insulinogenic response, which works to the insulin receptor and then activates this intracellular signaling pathway that is involved in activating the initiation of translation towards polypeptide and then muscle protein synthesis. Over here, we know that simply just having protein in our diet and the amino acids from that protein, we know that they all have specific amino acid transporters. And so they have the ability to enter the cell leucine, it shows here simply by its sodium-independent transporter, it enters as well, and it shows here that, you know, all amino acids here are contributes to the amino acid pool within skeletal muscle, which obviously those amino acids, all those the proteogenic amino acids are going to be required for muscle hypertrophy because of their binding to cognate charged transfer RNA that participate in translation. But leucine specifically, you know, data shows it interacts not only with the pool, but also specifically with mTOR, which is involved in this mTOR pathway that's involved similarly with what we see with insulin signaling in initiating muscle protein synthesis. So the general process here is that, based on what we just talked about of instigation, this just kind of is trying to show all of these processes, if you will, are somewhat interrelated and integrated in a complex way, and all work hand in hand together to help support muscle hypertrophy. So think about it relative to the fact that with resistance exercise, obviously we get the mechanotransductive component. We can also get the impact of the hormonal component as well, whether or not somebody is utilizing growth promoting hormones that are exogenous. And then of course the nutritional component cannot be overlooked, because, you know, we can go to the gym all we want, but if our diet's not right and we're under eating and providing, you know, a hypo nutrient balance, then that's going to then impact how much muscle hypertrophy that we hope to enjoy. So, you know, this nutritional component is obviously one that's extremely important. So anyway, I've just had this on here just again show the fullness, if you will, and the complete orchestration of all three of these factors that are involved in instigating this process for muscle hypertrophy. This slide, I just have one slide here, just, you know, in terms of interaction, that's the next. Remember, it's an instigation, interaction, then we'll go to transcription. Again, interaction is just the process by which the stimuli, the instigative components, are linked, if you will, to the transcriptive process in the gene. So we have this interaction between the stimuli and gene expression. And there are a number of cell signaling pathways, and these are just two very common ones that we see in an anabolic related scenario. And so this just shows that this is receptor tyrosine kinase signaling, whether it's insulin, IGF-1, growth hormone. We have the ability to upregulate this AKT-mTOR pathway, which we just talked about a little bit on the previous slide, that's involved in initiating a translation towards protein synthesis. But the interesting thing with that is that we also have crosstalk with a pathway called the MAP kinase, mitogen activated protein kinase pathway. Specifically, within that, one called the ERK, extracellular regulated kinase pathway, that is involved as well. And so, interestingly enough, the ERK pathway specifically upregulates gene expression. The AKT-mTOR pathway specifically plays a role in initiating and helping to upregulate translation for muscle protein synthesis. So it's very, very important that these two pathways talk to each other, because we can't have one without the other. I mean, if we're ramping up for muscle protein synthesis without a pathway that is providing ample amounts of substrate messenger RNA, then there's nothing there to translate. So this pathway here is being interactively activated to help facilitate muscle-specific gene expression. Those genes transcribe themselves and provide the messenger RNA as template for translation that is going to be initiated by this pathway here. All right. So this is the interactive component. This is here. There are some pathways that work against this process, and I'm not really going to get into that. Actually, that could be a whole nother lecture all in itself. But there are some cytokines that are, you know, some pro-inflammatory cytokines that could be elevated during times of systemic inflammation. heavy oxidative stress that actually could work against this process. So very quickly, when we talk about transcription, transactivation of genes, we know that this is kind of an anatomy of a gene. We have this area downstream of the promoter that contains the coding region that's going to give rise to the amino acid sequences that end up being involved in a particular protein. And then we have the core promoter and promoter regions, and these are the ones that where the gene is actually initially turned on. This is like the ignition switch. And then the proximal control elements and then regulatory control elements, even more upstream, if you will, are like the accelerator pedal. They provide a way of actually upregulating and providing even more activity to that gene. And this is just kind of a cartoon, if you see a process for how genes are transcribed. You've got the basal transcription factors here at the core promoter, and then we had the proximal control elements through the CAT box and the GC box that are involved in linking our basal transcription factors to our regulatory transcription factors. These are the ones here that can significantly increase the expression of an inducible gene, not a constitutively expressed gene, but an inducible gene. And we can also have repressors or silencing sequence in here that actually are involved in slowing or down-regulating the expression of a gene. This just shows very quickly that genes are categorized as either stress-responsive genes, metabolic priority genes, metabolic mitochondrial enzyme genes, meaning that all genes do not express themselves based on the same timetable. Some are very quick to respond in response to a stimulus. Again, stress-responsive genes. There are some genes with resistance exercise that are expressed very quickly within minutes of the stimulus and are short-lived. There are some that are more intermediate response genes like these here, and there's some that are more latent in terms of the reactivity to the stimulus. But that being said, this just shows that for the most part, after about 24 hours, then the activity of any of those genes in response to a stimulus, for the most part, have pretty much run their course. So translation is just the process where when we've created a mature messenger RNA that's ready to be read, READ, or translated, then this process with which we'll use amino acids bound with transfer RNA will interact with the ribosomal network that associates itself on the mRNA and ends up then translating or then decoding those nucleotide sequences and replacing those sequences with amino acids. So we go from a nucleotide strand to an amino acid strand, and in so doing, we synthesize a polypeptide. That polypeptide in time, as it begins its process, will then become biologically active, if you will, so that it can be incorporated within the cellular matrix and used accordingly. This simply is just an overview of protein synthesis, where again we just talk about the instigation, whether it's nutritional or hormonal or mechanotransductive, through interaction upregulates the expression of genes, leading to transcription and then translation and then the overall synthesis of a protein per se. All right. So this is where I was talking about the other category on the first slide of cooperation, and this is one that it just continues to just fascinate me more and more. We're learning more and more all the time about just how satellite cells behave. Satellite cells are simply just muscle stem cells. They're like baby muscle cells that never developed. They're sitting here lying quiescent along the surface of a skeletal muscle, if you will, and in certain scenarios can activate these satellite cells. We know now that heavy load exercise, particularly that is enough load to contribute to muscle damage, will actually call these satellite cells into action out of quiescence. And then there's a process here that they go through where they proliferate and they differentiate into myotubes. And then they donate their nuclei. They're not contractile, but they donate their nuclei to pre-existing mature muscle fibers. So what ends up happening now is the muscle fibers, as a result of satellite cell activation, now end up having more nuclei. If you have more nuclei, you have more DNA. If you have more DNA, you have more genes to be expressed. You have more genes to be expressed, then you generate greater power for muscle protein synthesis, if you will, based on an enhanced transcriptive scenario. So that's just what this slide shows, where we have no hypertrophy. This is a satellite cell, and it's showing this is a myonucleus, and this is just an unstimulated muscle fiber. Over the course of time with resistance exercise training, the muscle can hypertrophy, and it can do so quite significantly, but it can do that without the significant assistance or any assistance from satellite cell activation. So this means this is just hypertrophy that occurs just strictly and purely from just enhanced muscle protein synthesis. But there comes a time where satellite cell activity ends up coming into play. Again, we're finding out more and more all the time about the different scenarios and the time course, if you will, of when satellite cells actually come into play. There's a study that was done a few years ago that estimated that a muscle can hypertrophy up to about 25% of its pre-hypertrophy amount, and to hypertrophy any above that would require satellite cell activation. Now, I've seen some other data that refutes that a bit, but still there are constructs involved in helping to define and delineate how satellite cells come into play. And so if that's the case here, beyond simply just purely muscle protein synthesis without the assistance of satellite cells, once these satellite cells end up running their course, they donate their myonuclei, and again, now we have the muscle fiber has the ability to hypertrophy even more, because as I said earlier, it has now more genetic, I guess you'd say, power to be able to do that, because it has a greater amount of myonuclei. So this is really a cool figure that it was adapted from Brunsgaard back in 2010. And then Dr. Jimmy Bagley, who's at San Francisco State University, actually made this, and it's I've used it quite a bit in class, and it's pretty cool. But it comes into play relative to satellite cells with hypertrophy and this mechanism of muscle memory. We all know, those of us that have trained, that we can train muscle, it can hypertrophy. Then if we detrain, then obviously muscle atrophies. And if we go back and we retrain, then typically that muscle will hypertrophy at a much greater rate than it originally did when we first started. Well, why is that? Well, now it seems like that satellite cell activity that occurred during the training process that in donated myonuclei, once we retrain, those myonuclei are retained. And so then when we begin retraining, then again, because we have this greater amount of genetic power to be able to induce muscle protein synthesis, that's due to the satellite cells. This is my last slide, and just simply, and I'll hit this real quick, and this is the issue of degradation. This easily is another webcast all itself, relative to some of the constructs relative for inducing muscle protein degradation and impeding hypertrophy. So we know real quickly, if we have decreased amino acids in our diet, for instance, our nutritional protocol is just off base, that has the ability to impact muscle protein synthesis in a negative way. Other scenarios relative to hormonal imbalances or something that might be going on otherwise that might have reductions in some of the hormone growth factors, IGF-1, growth hormone, for instance, testosterone, those of us that are no longer young pups anymore, we deal with decreased testosterone. So that plays a role as well relative to sarcopenia in us as we age. Other factors that come into play: systemic inflammation, reactive oxygen species that can occur from various disease states, inactivity, just poor nutrition, if you will, can increase the levels of circulating pro-inflammatory cytokines. Tumor necrosis at factor alpha is a biggie, and it will play a role in upregulating oxidative stress and cellular apoptosis. And then we also know the negative regulator of muscle mass, myostatin, if it's increased for any reason at all, particularly during certain scenarios of catabolism, it certainly can have an overwhelming impact on on the maintenance of muscle mass as well. So, final thoughts: muscle hypertrophy, guys, obviously is a process that occurs over time in conjunction with muscle overload. Of course, muscle overload, like Dr. Roberts will talk about, that's a way that you can incorporate a number of different scenarios that can impact hypertrophy. Of course, it's contingent on a state of anabolism. This is what my last slide kind of alluded to. We need to try to maintain more of an anabolic state where we're synthesizing more protein than we're degrading. If that's the case, then we have positive accretion. And then obviously, like I said, hypertrophy is associated with a conglomeration of cell signaling pathways. They all, it's highly unlikely that any one is going to be any more important than the other. They really all have to operate synchronously, if you will. All right. And down here, the last one is that muscle hypertrophy, without a doubt, is critically linked to a sound nutritional approach that without a doubt involves adequate amounts of protein. So, with that being said, I thank you. I thank the ISSN for hosting this webcast, and for those of you that are here and your attentiveness and the support of Dr. Antonio and Dr. Roberts, and thank you very much.
Hey, Dr. Willoughby, that was great. And it's interesting. I used to, way back when I was in grad school, as you know, I had a huge research interest in skeletal muscle hypertrophy.
Yeah, and satellite cells. I remember. Yeah,
right. Satellite cells.
You're working with Dr. Gagne. He's the pioneer, literally.
Yeah, we're literally talking 3 decades ago. But what's interesting is that the more you get into it, the more obviously it's much more complicated than even when I studied it way back when. And I think you hit on a really pragmatic point, and this will be a good segue to Dr. Roberts. Is that at the end of the day, you have to train, and that training is the primary stimulus. No matter how good your nutrition is, it's still primarily training. I mean, there are people who train who eat like crap and they still grow. You could eat perfectly and not grow at all. So again, training is sort of it's the paramount consideration. So what I want to do now is transition to Dr. Roberts, because he's actually going to go over the skeletal muscle hypertrophy in response to different load schemes. So this might be more on the pragmatic side, whereas you covered a lot of the molecular biology of it. And Dr. Roberts is currently at Auburn University. He does some super cool stuff on skeletal muscle adaptation and plasticity. So without further ado, let's have Mike share his screen and edify us.
Sure. Can you all see the proper screen? Let's see. Is that okay?
So far, we do not see your screen.
Perfect. Let's see. Share screen. Oh, there we go. Okay.
Let's see.
How are we doing?
Perfect.
Okay.
Yeah, great.
Well, thank you for the brilliant talk, Dr. Willoughby. It's always a pleasure to hear you and refresh my knowledge on a lot of that stuff. I remember sitting in your class when you were teaching PhD students. I was a master's student, and I think I had asked you, can I audit this class? And you were gracious enough to let me do it, and you really lit my fire. So I just, every time I see you, I'm always thankful for the opportunities to work in your lab and learn from you. So yeah,
I appreciate that. You were a blessing then, and you continue to be that way. So thank you.
Thanks. So first, I'm gonna see here. Okay. I do want to disclose that we have a lot of different funding streams. I predominantly do work for the industry. So there's that. I have been a paid consultant in the past. I don't own any stocks in nutritional supplement companies, and I don't even know which stocks I do own in my retirement account. Now, what I want to talk about today is this paradigm true regarding strength training versus hypertrophy training? We've heard a lot, you know, in the past decades that if you want to get as strong as possible, you want to lift heavy weights for low repetitions. You want to make sure that your bioenergetics are sufficiently recovered. So that means you want to take a lot of rest in between sets. And then you want to do compound movements. This differs a little bit from hypertrophy training, where this is more higher volume training, right? If we're lifting as a percent of our 1 rep max, maybe 85 to 90% 1RM with strength training. Hypertrophy training can take a lot of forms. Typically, people think about this in the 70 to 80% 1RM range, although we'll talk about some newer paradigms that are emerging where people are training with very light resistance at 30% 1RM to failure. And you want to focus on doing isolation movements along with the compound movements. What I want to ask today is, does the current evidence back these paradigms up? And if it does, you know, is this simple strength training with higher resistance and lower volume, hypertrophy training, you know, with higher volume and lower resistance, right? Is it as simple as that? So when I talk to students and when I give conference lectures, I typically define high load training as doing something like 4 sets of 3 at 90% 1RM, although clearly different iterations can exist. I define moderate load as something like 4 sets of 12 at 75% 1RM. And low load, very high volume training would be something to the tune of, you know, 4 sets of 30 repetitions at 30% 1RM, right? Now, if you read the NSCA's text, they basically have one of their training principles, which is training specificity. And to summarize that principle is to say that if you want to get stronger and hypertrophy to a greater extent, then you should lift heavier weights, right? Well, how do researchers like Dr. Willoughby, our laboratory, and others test this hypothesis? The idea, for those that haven't been involved in these studies, is that we typically set up a pre-testing battery where we get body composition, muscle biopsies. We can get localized hypertrophy measures with an ultrasound device. And then if you want to test the high load versus low load paradigm, you would have 2 groups or 2 cohorts, one which trains with high load training and the other with high volume training. You know, this can take form, this can last 6 weeks, this can last 8 weeks, 12 weeks. You typically don't see studies longer than 12 or 16 weeks because a lot of these studies are done on college campuses and they're limited to the academic calendar. And then after the training paradigm concludes, you typically wait 48 to 72 hours and you perform your post-testing battery, which again involves body composition, ultrasound, strength assessments, muscle biopsies, etc. Okay, so that's kind of how the research works. There are various methods used to determine if muscle hypertrophy occurs. And I just want to talk about those real quick because I'm going to be showing you all a lot of bar graphs and data points related to these measures, right? So MRI, anytime you read a paper that uses MRI, you typically get a cross-sectional view of the thigh muscle. Some studies have done the arm muscles, and you can quantify the amount of meat that's on the bone, so to speak, right? And you can say, well, if the pre-score was this and the post-score had more skeletal muscle, then we can quantify that in terms of percent change in muscle mass at mid-thigh, okay? Same thing with PQCT. We actually have one in our lab. Very good device, doesn't provide the resolution like an MRI, but the same idea. You can do a mid-thigh scan, get changes in muscle area. Ultrasound's a little bit different. Now you can do panoramic ultrasound to look at changes in muscle area. We just do transverse snapshots to get muscle thickness at the vastus lateralis. As it relates to muscle biopsy measures, typically fiber CSA is king, and you can do fiber type specific CSA. You can quantify the change in type 1 fiber cross-sectional area or muscle size, as well as type 2 fiber cross-sectional area. And then what's not commonly reported, but needs to be, in my opinion, is using methods such as TEM imaging, because this allows you to look within the cell and look at changes in cellular structure with different training paradigms. All right, so what does the data say about high load versus high volume training? Well, this is one of the studies that first addressed this question. It's about 20 years old. It's been cited a lot, over 1,000 times, by Will Kramer and Bob Stearns' group. And the gist of the study was they had 3 groups of college-age men, which did 3 forms of training. One was high-load training. This means that every exercise that was performed was training at 3 to 5 repetition maximums. Okay, so that's heavy weight, low reps. They had an intermittent load training group, trained at 9 to 11 RM, and then they had a high volume training group where every set consisted of 20 to almost 30 reps, okay? I'm only gonna present data with high load and high volume. I made the graphs to include those and not the second group. Exercise was done 2 days per week for 8 weeks, and it included leg press, squat, knee extension. Okay, they took pre and post leg biopsies and strength assessments. Now, this is the histology data looking at the muscle fiber size, right? Type 1 fibers, these are your slow twitch muscle fibers. You can see that the high load training significantly increased type 1 fiber CSA, numerically increased type 1 fiber CSA with high volume training, but that wasn't significant. And then looking at type 2 fiber CSA or type 2A fiber CSA, you can see that high load training, again, facilitated an increase in muscle fiber size. High volume training did not, okay? So far we can say that high load training is more hypertrophic in terms of the cellular adaptation. This is the just one of the strength measurements that they obtained. This is leg press. And again, comparing high load to high volume training, you can see that those that trained with heavier weights got stronger compared to high volume training, right? Both groups increased in strength, but high load training facilitated the greater increase. After that paper was published, Andy Fry published a very nice comprehensive review article. And again, the gist of the article was if you train with higher loads, this is based on 4 studies, you typically see the greatest increase in 1RM strength based on leg press and leg extension. If you train with loads around 30 to 40% 1RM, you can see increases in strength, but they're not as robust, right? He then presented data from around 6 studies looking at type 2 muscle fiber hypertrophy, and the theme was the same. Heavier loads elicit a greater increase in cell size versus lighter loads, right? So, case closed, right? If you want to get bigger, stronger, potentially faster, you train with heavy weights, don't mess with this high volume stuff, correct? Well, that was true until 2012. Stu Phillips really stirred the pot, for those that have seen this paper by Cam Mitchell. And what he did was he had individuals that did leg extensor training under 3 conditions. The first condition was 3 sets, 30% 1RM to failure. Second condition was only 1 set, 80% 1RM to failure. Third condition, 3 sets, 80% 1RM to failure. Okay, training was 3 days per week for 10 weeks. Limitation of the study was it's only leg extensor training. They didn't, trained with compound movements, but nonetheless, very well-controlled study design. They did pre and post strength assessments as well as mid-thigh MRI scans and biopsies prior to training as well as 48 hours after training. And this was really the one of the more profound figures from the study, and that is to say, those that trained at 30% to failure using 3 sets of leg extensor exercises per session saw a similar increase in muscle area or quadriceps area compared to those that used heavier weights, right? So as long as you did 3 sets of one or the other, you saw similar increases in muscle hypertrophy. If you only did 1 set at 80% 1RM, you saw hypertrophy, but it wasn't as robust, okay? Also looked at leg extensor 1RM strength. Now this is showing that all groups increased strength. Those that trained with 80% 1RM saw a little bit greater increases in strength. However, when looking at an isokinetic dynamometer, peak torque during knee extensor, you can see that there were similar peak torque adaptations. So by and large, this study is saying as long as you train to failure, whether it's with lightweight or heavyweight, you can see similar hypertrophic adaptations and basically similar strength adaptations, right? What I'm not showing is the histology data, but they reported that both groups experienced similar increases in type 1 and type 2 muscle fiber hypertrophy. Again, this stirred the pot. I can't impress that upon the audience enough if y'all haven't kept up with this area of the literature. So really to summarize this paradigm, if I train this way with the neon colored dumbbells at the gym, as I like to call them, if I train with heavy weights, right, will I experience similar training adaptations so long as I perform either to failure, right? Well, I'm going to present data that says maybe, maybe not, right? So Nate Jenkins, he did this when he was with Joel Kramer at Nebraska. He sought to basically replicate Cam Mitchell's data, had 2 groups of college-age males, one that did 80% 1RM leg extensor training, one that did 30% 1RM leg extensor training. This was 6 weeks of leg extension training, just like Stu and his group did. They performed 3 sets per training session, 3 training sessions per week, and they did some neuromuscular tests at the pre and post testing batteries as well as ultrasound for muscle thickness. A little bit of a busy slide, I apologize. What's interesting is if you look at the total repetitions performed over the duration of the study, clearly if you lift lighter weights, you have to do almost 3 times as much lifting. There is more time under tension because you are lifting for more repetitions. But what is interesting is that the training volume almost comes out to be identical whether you lift 30% 1RM or 80% 1RM, so long as it's to failure, okay? Now this is the muscle thickness data, right? This is pre-training, this is midpoint training, and this is post-training, right? This is the group that performed heavy lifting. This is the group that performed light lifting. And what you see here is that muscle hypertrophy indeed was similar between groups. This is vastus lateralis, so the outer muscle of the upper thigh. And so this sort of follows what Stu's lab argues, and that's whether you do light or heavyweight training to failure, you are going to see similar hypertrophic adaptations. This is the strength data in terms of looking at leg extensor strength, and clearly, right, those that trained with heavier weights saw this continued increase in strength. Those that trained with 30% 1RM saw a little bit of an increase, but it was significantly less than heavy training. And I thought this was a nice contribution to the study question, and this added data in terms of what Stu and them didn't measure. Using twitch interpolation, they showed that pre to post training, you can actually activate more muscle fibers or motor units with heavier training. So the neural adaptations and the neuromuscular adaptations facilitated seem to be greater with heavy training versus light training, okay? Around this time when Nate published his paper, we sort of got involved with the study question. Cody Haun really is what drove this interest. He's now with his own company, former student of ours. So I'm going to talk about Cody's data, and then I'm going to talk about a chronic training study that followed up by Chris Van, who's currently in the lab. And we're doing this in collaboration with Stu Phillips. So Cody's study, we had 15 college-age male participants. Study design was a pre-muscle biopsy. They did 4 sets of failure at 30% 1RM, 15 and 90-minute post-biopsy, and then 2 days later, we did biodex. And by the way, we did biodex at pre as well. Okay, so we have a lot of biopsies here, and then we have a pre and 2-day post biodex with light load training to failure. They took a 5-day break, and then we replicated the same exact testing procedures, but this time 80% 1RM training to failure was implemented, right? So this is one acute bout or one bout of exercise looking at the acute response. Just to show you all, this is the number of repetitions performed when people did 30% 1RM training to failure versus 80%. Clearly, you do a lot more repetitions with 30% 1RM training to failure. On average, the first set consisted of 70 leg extensors, which seemed quite painful when we were monitoring these participants. They did not enjoy that training protocol, right? And this is the volume load. So what is interesting is this sort of agrees with Nate Jenkins' data. Even though you do more repetitions, many more repetitions with light load training, at the end of the day, looking at the total volume load or total weight lifted, it seems to be roughly equivalent, right? Whether you train to failure heavy or light, you're going to lift the same amount of weight, typically. Now, busy slide, right? This is the biopsy data. I just want to impress upon y'all that we looked at proteolytic markers, and Dr. Willoughby touched on that. That's important for the growth response. So we looked at those at the mRNA level. We looked at some pro-inflammatory cytokines. We looked at PGC1 alpha, which is sort of the master regulator of mitochondrial biogenesis, and then we looked at mechanogrowth factor mRNA levels. We also looked at phospho-signaling targets in the mTOR1 pathway, which Dr. Willoughby talked about. Long story short, I'm not going to go through each graph. I'm just going to say that light load training and heavy load training showed the same molecular feature, meaning that all of these markers were similar in terms of the response to each form of training. So that's interesting, right? That could indicate that indeed there could be similar molecular adaptations, whether you're light load training or heavy load training, okay? This for us was really the most important finding, and that's looking at the biodex data. Now, the way we're expressing this on this slide is the percent change from pre-training levels. So remember, we did this at pre and then 48 hours following the exercise bout. We saw with 30% 1RM training at each knee extensor speed, there was a significant decrement in the amount of torque that was able to be produced. When you compare that to 80% 1RM training, yeah, we saw a decreased 2 days post, but it wasn't as robust as high volume training, okay? And so really to summarize in terms of what we learned from this study, looks like at the molecular level, there are a lot of similarities with light load training, 30% 1RM to failure, versus heavy load training. That agrees with Stu Phillips' lab, his findings, right? In order to get the similar response, though, if you want to go down if you want to do light load training, you have to train to failure. This typically involves 30 up to 70 repetitions per set. Very time consuming, very exhausting. You get the burn, it's not pleasant, right? If you're training with 80% 1RM, on leg extensors, this typically involve 10 to 20 reps per set. With compound movements, it's probably more on the tune of 8 to 12 reps per set, okay? This is the key finding, and that's training with light weights to failure. When we looked at that biodex data 2 days after the training bout, led to more residual fatigue versus training at 80% 1RM to failure. I'm going to talk a little bit more about that later on in the presentation. Now, this is our second study by Chris Van. He got 15 college-age male participants that were well-trained. We did MRI, ultrasound, muscle biopsies, strength testing, okay, on one leg. Same thing on the other leg. Now, one of these legs, participants did single leg press training and leg extensor training 3 days per week. This is week by week breakdown in terms of sets and reps per week for both the leg extensor and leg press exercises. And this is the training load, right? We wanted this leg to be the high load training condition for 6 weeks. This is the high volume leg, right? So again, week by week, you can clearly see higher volume, 60% 1RM. What we liked about this study design is that each participant was his own control. And when we looked at pre and post change scores and all of these things that we were measuring, right, we can confidently compare higher load training to higher volume training. And then after the training, we replicated the same testing battery, doing MRI, ultrasound, and muscle biopsies. Now we're working on the MRI data now, but we do have the ultrasound data. It seemed like the higher volume leg facilitated a little bit more of an increase in vastus lateralis muscle thickness. Now each one of these squares and lines is an individual participant. I'm just summarizing the percent change from pre to post here. You can see that there was a 6% change in the high volume leg, which was significant. There was a numerical increase, but that was not significant in the high load leg. So far, high volume training, at least according to the ultrasound, facilitated more of an increase in hypertrophy. However, when looking at strength, this is leg extensor strength, both training forms similarly increase strength, okay? So this sort of disagrees with Nate Jenkins and Stu's findings. And we say if you train 60% 1RM, or if you train around 90% 1RM, you actually see similar increases in leg extensor strength. This should be, sorry, I'm going back and forth. This should be leg press strength. We also did unilateral leg press strength. And clearly you can see the same trend, that there were similar increases in leg press strength with either higher volume or higher load training. So to summarize what I've talked about so far, the evidence suggests it looks like I can train with whatever loads I want, lighter loads or heavier loads, so long as I train to failure. Is that the case? Not so fast, my friend. And the reason I say that is, first, should one be training to muscular failure in the first place? Kevin Carroll, he was a PhD student in Mike Stone's lab. I thought they published a very nice study recently, and they compared the training adaptations between two groups. One group trained to relative percentages of 1RM, but they did not train to muscular failure. Okay, so that's this RISR group. The RM group trained every exercise to muscular failure, trained 3 days per week for 10 weeks. These are their data. So this is type 1 muscle fiber CSA, or muscle fiber size, type 2 muscle fiber CSA, or size, and then this is ultrasound vastus lateralis muscle size using that panoramic ultrasound method. Clearly, if you don't train to failure, you see that you observe greater or experience greater increases in muscle fiber size as well as muscle size. If you train to failure, you don't see that, at least according to Kevin's data, right? And this is sort of paraphrasing the author's own words in the discussion. They say previous researchers have suggested training to failure permits the maximal recruitment of all motor units within a task. So the translation is, we used to think training to failure was a good thing because we recruit all of our motor units and muscle fibers. However, recent data suggests that performing bench press and back squats to failure delays recovery of neuromuscular performance, days after training. And so the translation, which we agree with, is if you always train to failure, you're not allowing your motor units or your muscle fibers to recover. And so if you have a training block where you go week to week to week training to failure, this may be the reason why you don't see as much muscle hypertrophy, okay? And this could be, you can call it overreaching, you can potentially call it overtraining, but the idea is that you don't always want to train to failure, right? Whether it's with light loads or heavy loads, okay? And then second, and this is one of the last points I'm going to bring up, people tend to really look at a few variables and overemphasize those variables while not looking at other variables and saying, you know, okay, our fiber CSA increases with light load training and heavy load training the same, so that means training methodologies are the same. You can do either one of them, right? But there are other fine details, and that's one thing our lab is really starting to look at. So I'm just going to go over these studies, and I know, Joey, I apologize for the time. This will take 2 minutes. We recently published 2 studies within the past year. One study was extraordinarily high volume in previously trained participants. They did 6 weeks of resistance training at 60% 1RM. By the very last training session, if you were in the study, you had to come in and do 12 sets of 10 squats, as well as 12 sets of 10 deadlifts, and then you had to do the same thing for some upper body exercises. It was a very painful study to watch. I do not recommend trying to replicate this study. I thought I would have to go to the IRB and explain all these injuries. Very painful to watch. Our subsequent study, we did high load training. Now, instead of 6 weeks, it was 10 weeks where participants train 80 to 90% 1RM, okay? I'm gonna show you some of the data from both these studies. The first study, very high volume training. What's interesting is we didn't see a pre to post change in muscle fiber CSA. Our whole body DEXA data suggests that if you just look at DEXA lean soft tissue mass, you know, from pre to mid to post, you do observe an increase in muscle mass. But if you correct for extracellular water, which we think is edema, potentially muscle swelling from non-contractile elements, right, you see an increase from pre to mid, but not necessarily mid to post. So high volume training, in our opinion, may involve some fluid shifts that people don't commonly talk about. And then finally, we used bottom-up proteomics, and we showed that in a subset of participants, a lot of glycolytic enzymes as well as creatine kinase was upregulated at the protein level. And so we viewed that metabolic conditioning may have resulted from higher volume training at the molecular level. In the second study, we did see with high load training about a 20% increase in type 2 fiber CSA, which was significant. We didn't see an increase in type 1 fiber CSA. We saw an increase in strength. And what's interesting is that we saw a decrease in whole body fat-free mass by 1 kilogram, which was significant, okay? Again, we did a bottom-up proteomics from, you know, pre and post muscle samples. And what we discovered was that metabolic conditioning, or the upregulation in glycolysis enzymes and creatine kinase and mitochondrial enzymes, did not occur with higher load training. So higher load training doesn't seem to prime the bioenergetic systems in terms of upregulating enzymes needed for those processes, okay? So our interpretation of the data is with higher volume training, there's possible fluid shifts or fluid retention in muscle compartments. This may account for some of the increases in muscle mass. High load training, there's possibly fluid shift outside of the muscle, which accounts for minor decrements in fat-free mass. We think this is something people need to start paying more attention to, okay? Proteomics data suggests that metabolic conditioning occurs with higher volume training, not higher load training. And then again, this is Kevin Carroll's data and Cody's data. If done excessively, we think that higher volume training can lead to more residual fatigue. And we think that higher load training, not so much. We also think that higher load training facilitates more of an increase in strength, even though some of our data doesn't suggest that. Okay, this applies to previously trained folks. If you're untrained, it probably doesn't matter. Okay, you can train high volume, high load, whatever. You're gonna get stronger, you're gonna get bigger. As you become more and more trained, these variables become more important. Okay, so the debate continues. I'm just going to show you that there's a lot done here in this space. Some people say high volume is better than high load. Some people say the other. And so there's that, okay? Okay, so how do I train again? Well, to sort of summarize our data, we think when not training to failure, if you want to get bigger, if you want to get stronger, train heavier. Train 70, 80, maybe even higher percent 1RM. If you want to mix up your routine, that's where we think that training, you know, implement maybe during a 4-week or 6-week training block, you can have 1 or 2 sessions for each muscle group where you sort of shock the system with higher volume training, maybe 30% 1RM, right? And then don't train to failure all the time, whatever you do, because again, the data is starting to come out that leads to suboptimal training adaptations. So with that, I'll thank ISSN, Dr. Antonio, again, Dr. Willoughby. You lit my fire. Man, this was 15 years ago. So I have you to thank for sort of introducing me to this space, as well as Rick Kreider, Chad Kurtzik, and Frank Booth. They've all been prior mentors in the past. I have a lot of students that do this work, so I wouldn't be talking without their incredible work. We have a lot of collaborators. In particular, Stu Phillips has been very kind to us, and we're working on some projects with him in this area right now. And then I want to thank all of our funding sponsors for that. And with that, I guess I'll take virtual questions.
Hey, Mike, that was an awesome presentation. It's you know, I met I don't know if you followed bodybuilding way back when, but one of the first bodybuilders to visit me in the lab when I was a grad student was a guy named Lee Labrada.
Yeah.
And Lee, and I'm sure Darryn is familiar with Lee, obviously he was interested in muscle hypertrophy. And even back then he said, if you look at how all these guys train, we all train differently. And in a way, the data is supporting how there's such a wide variety of, I guess, training paradigms or protocols in terms of sets and reps and whatnot. So one of the attendees had a question, which I think you sort of answered, but perhaps elaborate on it. He wanted to know if there's a set number of reps that you can say will maximize muscle growth. You know, let's say do 100 reps per muscle part or body part, or 200. And I think you alluded more to volume than to reps, but address that for us, please.
Yeah. I think so. The more we get into this, Joey, the more we don't know. And that is to say, if you take an ultrasound probe and you look at somebody's, you know, change score from pre to post training after a higher volume block using 60%, you know, 1RM training versus 85 or 90% 1RM training, perhaps the higher volume training does increase muscle thickness, right? However, what we think is there could be more that's going on beyond the addition of contractile protein. Like I alluded to, some of Cody's data shows that maybe fluid retention in the muscle comes into play. You know, one analogy I like to talk about is, what's that?
Oh, nothing, no.
Yeah. Arnold Schwarzenegger in Pumping Iron used to talk about the pump, right? When you did this sort of high volume training. And I think, you know, that could again be fluid shifts, and that could explain why you see this enlargement in muscle. Now there is going to be some myofibrillar protein synthesis occurring, you know, following exercise, and there's going to be the addition of myofibrils. And eventually an increase in fiber CSA. But what we ask and sort of have posed is, you know, is there also fluid shifts, meaning this you know, extracellular to intracellular fluid flux that contributes to the increase in muscle fiber size when you do higher volume training, right? That's to be determined. I don't have a good answer for that. That's just sort of our working hypothesis right now.
Yeah. I would add, and that's Mike. That's definitely a good explanation based on what we know and we don't know. But you know, we also are finding out more and more about this notion of time under tension, and
yeah.
You know, the longer muscle stays stimulated, of course, you know, the greater capacity is for the upregulation of all these intramuscular elements that are involved that I talked about and you've talked about, or contribute to ultimately hypertrophy. But you know that you know, you're talking about time under tension, but load also ends up coming in to be somewhat important as well. But then again, there's another issue of, you know, how long should that muscle stay under tension before it starts to before the, you know, the metabolic influence starts to become, you know, impacted enough where that muscle is not able to, you know, maintain said said work requirement. So, you know, that's just, I don't know, that's interesting. And Mike, you've seen it. Joy, you've seen it. I mean, there are those people, you know, I've been bodybuilding for over 30 years. And so, I mean, I've seen people who, you know, who train with one style relatively, you know, with some brevity and just, you know, and I'm not necessarily talking about with the incorporation of pharmacopoeia because, you know, I remember, you know, many of these guys, my friends, were, this is back in the day when, you know, we were all just eating and lifting. But, you know, individuals that would train with a relative amount of brevity and then they would just hypertrophy quite well. And then others that, you know, it took a complete different training approach for, you know, for anything to even happen. And so, you know, there's just such an individualized approach here. And, you know, I don't know, you know, I don't know, Mike, what do you think? You know, all of a sudden you can talk about the, you know, the genomics of muscle hypertrophy, you know?
Yeah. We think, I mean, so, you know, I'm not gonna, you sort of want to pick a working hypothesis because, you know, I don't want to show a lot of data and then people ask, so what does it mean? And we say, I don't know. I just always thought that was an awful cop-out, you know, as a scientist. So even if I picked the wrong hypothesis, at least I, you know, stuck my neck out. So I guess in relation to this area that we're researching, one thing we need to do is look at the structural adaptations as well as the molecular adaptations more. You know, with proteomics, that sort of afforded us the opportunity to look at all of these various enzymes to get a more holistic picture of what's occurring. And then if you use things like TEM, right, you can look at the size and distribution of myofibrils. You can look at alterations in the mitochondrial reticulum, you know, with higher load versus higher volume training. So I think those sorts of studies would be informative as it relates to what you brought up, Dr. Willoughby, which is, you know, time under tension. How does that affect these variables, right? High load training. How does that affect these variables, et cetera? But as of right now, I mean, we're sort of taking stabs at it. I personally believe that if you implement higher load training, like 30% when in training to failure, you'll see increases in fiber CSA, but I think a lot of that's due to fluid shifts. That's where I stand. Stu adamantly disagrees with that. So that's something that we, you know, we've solicited his expertise to help us with Chris's data, and we'll see how that shakes out. But you know, Cody's data, that whole body DEXA data, when you account for changes in extracellular fluid using the BIS device, it looks like that as you train with more and more higher volume at the whole body level, there's this hanging on to extracellular fluid, which we think is edema, right? I mean, and if there's edema, there's going to be fluid shift from the outer to inner portion of the cell as well. So that's, you know, that's where we stand. We think that high volume training with low load, the mechanisms of hypertrophy are you know, maybe the same if you look at certain biomarkers, but if you look at these changes in ultrastructure and you look at these changes in protein content with proteomics, you may see something that's a little bit different.
Well, gentlemen, I hate to end this party. We're out of time, and besides, I'm really hungry, and I'm sure Darryn wants to eat his fourth meal. But for those of you who may have missed the beginning of the webinar, it'll be it's being recorded and eventually be posted. So just send us an email through the ICSA website, and I'll send you a link to the webinar. And I want to thank Dr. Willoughby and Dr. Roberts for edifying us on skeletal muscle hypertrophy. And hey, hope you guys have a great rest of the day.